In this video, we are going to talk about the mechanisms shared by all serine proteases. And serine proteases are enzymes that catalyze the hydrolysis of peptide bond in proteins and peptides. So all serine proteases share the same three amino acid residues in their catalytic or substrate binding sites. And those three amino acid residues are serine, histidine, serine is protonated at first, serine, histidine, and aspartic acid, and together they are called catalytic triad. Catalytic triad. So the mechanism involves nucleophilic attack of this oxygen of serine side chain at the electrophilic carbon of the peptide bond. But when the side chain of serine is protonated, oxygen is not as powerful nucleophile as is needed for this reaction. And it's kind of difficult to deprotonate it because the pK of this hydroxyl group is higher than 14, so it's not easily achievable under physiological conditions. But we have here some help from some other members, some other side chains of amino acids present in the active site. So histidine would serve as a base to actually abstract the proton of the side chain, but by itself it's not a strong enough base. So when the hydrogen bond between this hydrogen attached to one of the nitrogen atom of the imidazolium group of histidine is formed with the side chain of aspartic acid nearby, now it increases the basicity of the second nitrogen, and it can abstract this hydrogen, or I mean proton, right? And so the electrons are kind of assigned to oxygen for it to be negatively charged, right? But because everything is so close to each other, right, here in the active site of the enzyme, when already the substrate is present in the active site, those electrons can be simply used to form a covalent bond between this oxygen atom and the electrophilic center here in the substrate, which will be the carbonyl group of the peptide bond. And of course, not to violate the octet rule at this carbon atom, the pi bond between the carbon and oxygen needs to be broken. So as a result, what we'll have here, I will redraw the substrate. Of course, I will modify it accordingly, according to the transformations that we have shown here by those curved arrows. Serine will be covalently attached to this carbon. Histidine will be protonated. And of course, still hydrogen bonded to the aspartic acid residue. This intermediate is called tetrahedral intermediate. Because now it used to be sp2 hybridized carbon, right? But now this carbon is sp3 hybridized, and we know that sp3 hybridized arrangements of atom, sp3 hybridized atoms, carbon atom in general, would have a tetrahedral geometry, right? So what happens next? So another thing I wanted to mention that, of course, this is also called oxyanion, and of course it is stabilized in the active site of the enzyme because this negatively charged oxygen is in so-called oxyanion hole, which have two NH groups available to hydrogen bond the negatively charged oxygen, and those NH groups are from the backbone of the enzyme. and so the tetrahedral intermediate is stabilized. But what happens next? So next, this oxygen, which is negatively charged, even though it is stabilized as we discussed, but it would be willing to reform the pi bond. And so in this case, one of those two bonds need to be broken, right? Either carbon-oxygen bond, and in this case we just go back in the original state, or carbon-nitrogen bond. But nitrogen will have negative charge, right? And negatively charged amino groups are not good living groups. So it would benefit from protonation, right? Because if nitrogen is at least neutral, it would be better living group. So this protonation happens due to this histidine residue serving as an acid now. And so we will have the cleavage of this bond, right? And so we will have only the N-terminal fragment of the original peptide to still stay in the active site of the enzyme, now in the form of acyl enzyme intermediate. So serine residue is part of the ester bond now. And we have the second portion of our substrate released from the active site of the enzyme, right? What happens to the other members of the catalytic triad, right? So serine is here. So histidine, I will draw it right here. Histidine is back to its basic state, right? So here I have histidine, which is again in its basic state. And of course, aspartate is still here, right? So what happens next? Next, here we have water that binds in the active site as our second substrate. So water is right here. This is lone pairs. And of course, it can serve as a nucleophile, but it's, the nucleophilicity of water will be increased significantly if it is deprotonated. And we have a base here again. So if histidine here serves as a base one more time, so now the electrons from oxygen-hydrogen band can be used to attack our acyl enzyme intermediate. And we form another tetrahedral intermediate. So now we have serine still staying, and water attached, hydroxyl group is attached, right? And I'm going to erase, free some space here, erase the original state of the catalytic triad, because we are going to move in this direction, right? And so I have my histidine again as a base, right? So now I'm going to draw it right here. So I have histidine as a base again. And of course, it is still bonded to aspartic acid. And so, one more time, histidine serves as a base because, sorry, why is base? It should be an acid because it abstracted the proton. Otherwise, we will not be able to actually reconstitute the original state of the enzyme. So what happens now? Again, here we have unstable, even though stabilized by binding to oxyanion hole, unstable tetrahedral intermediate. So this oxygen and its lone pair of electrons here will tend to reform the pi bond, and so we are going to break one of the CO bonds. But again, this oxygen, if negatively charged, is not a good leaving group. So instead of assigning the electrons of this bond to oxygen, we are going to use those electrons to kind of take this proton, right? And this would regenerate our enzyme. So serine will go back to its original protonated state. Histidine will be back to its basic form, and we are able to release our second product, which will be finally the N-terminal fragment of the original peptide. So now it is bonded to OH group here, right? So that's the catalytic cycle shared by all serine proteases. And the examples of serine proteases are trypsin, chymotrypsin. So another thing that I wanted quickly to mention would be that in some enzymes, cysteine is used instead of serine. And in case of cysteine, so if we have cysteine here, we would have SH group. And so sulfur can serve as a nucleophile instead of this oxygen. But the pKa for cysteine side chain is only 8.5 or something, right? So histidine itself, without the help from aspartic acid, can deprotonate the thia group so that negatively charged sulfur is formed, which is a better nucleophile than just a thia group.